Hi, my name is Allison Leask. Welcome to my virtual presentation of distinct bacterial populations colonizing plastic debris in coastal waters of Southern California. The plastisphere is considered a unique ecosystem of microbes that attach to plastic in the ocean. Plastic pollution in the marine environment has been extensively studied. It has been proven that these microbes impact the ocean and its inhabitants. Microbial populations present on microplastics, tiny fragments resulting of physical and chemical breakdown of plastic debris. Little is known of the short-term impact of microbes attaching to plastic in coastal waters. This project has led us to focus on functional characteristics of microbial populations such as antibiotic production and potential plastic degradation. The genus Vibrio seems to be more prevalent on plastic and is of interest. This project evaluates microbes found on the following samples using live culture experiments and 16S barcoding: water, high-density polyethylene, low-density polyethylene, and polypropylene. Before plastic is deployed into the ocean, it must be sterilized by washing with ethanol and treated with UV light. All metal components are autoclaved. Plastic samples are zip-tied in a mini cage that is housed inside a larger cage. The color code resembles what type of plastic is within each cage deployed. The deployment analyzed his samples at different time points between 12 to 60 days. Deployment sites were located at Doheny State Beach in Dana Point, California, and Scripps Institute of Oceanography in La Jolla, California. In the left picture, a cage has been constructed and is ready to be deployed into the ocean. The right image depicts what cages look like when they are recovered from the ocean. Plastic samples have been deployed at different times of the year with average water temperature fluctuating. No clear pattern of colonization was observed over a month period. With longer incubation times, an increase in unknown, uncultured bacteria species was noted. Fluctuation of community makeup can be observed in the first 30 days. Culture media utilized in this project includes generic saltwater agar or marine agar and selective differential chrome agar Vibrio media. Plate A shows water bacterial colonization on chrome agar Vibrio media. Plate B shows polypropylene bacterial colonization on chrome agar Vibrio media. Plate C shows low-density polyethylene bacterial colonization on saltwater agar. And plate D shows high-density polyethylene bacterial colonization on saltwater agar. Cultures are observed and studied in order to determine what functional characteristics the microbes may present with. DNA was isolated from colonies using BioStick bacteremia kit. Amplification was completed using 16S primers 27F and 1492R. Sanger sequencing performed by GeneWiz allowed for sequence analyzation to be completed using DNA Subway. Phylogenetic trees were constructed using DNA Subway. This tree gives a visualization of the maximum likelihood between samples. The blue signifies samples deployed in Dana Point. The yellow shows which samples were deployed in La Jolla. Early in a deployment, there is no clear pattern based on location. Longer incubations show clustering among samples based on the location of deployment. A special interest has been taken in antibiotic-producing bacteria. So far, 6 distinct antibiotic-producing bacteria have been isolated from water and plastic samples. Colonies were patched on bacterial lawns of both gram-negative and gram-positive organisms. The presence of a zone of inhibition was considered a positive result. All have been proven effective against gram-positive bacteria such as B. subtilis and S. coni. All producers are alpha proteobacteria, mostly in the genus Phaeobacter. Fourier transform infrared spectroscopy was used to analyze the breakdown of plastic over time. Signs of oxidation of the plastic are apparent, especially in the HDPE sample. Signs of chemical breakdown are observed as early as 21 days of incubation. Scanning electron microscopy was completed in order to visualize the biofilms attached to the plastic on day 40. Pictured above are examples of each of the plastics deployed. This study indicates there are differences in the microbial communities growing on coastal marine plastic debris with potential implications for human health and the environment. After initial sequencing with only 27F primer, the samples were rerun to include both 27F and 1492R primers. This showed a more complete sequence. Chemical analysis needs to be continued on the antibiotic-producing microbes. Further testing can reveal the functional characteristics. Whole genome sequencing of the antibiotic producers needs to be completed for data mining and identification. I would like to thank my professors Dr. Anna Barral and Dr. Rachel Simmons for guiding me through this project. I'd also like to thank Dr. Jeff Bowman and his lab at SIO, Dr. Amelia DeForce, and National University students that are vital to the success of this project. I'd also like to thank National University and NSF for their support. Thank you.